I'm Dr. Claire Schubert, and I'm going to walk you through this poster on the characterization of BiTE-antibody-mediated cell killing using a combined live cell and flow cytometry workflow. The development of novel immunotherapies depends on the ability to quickly and effectively evaluate how well these drugs stimulate the immune system in order to destroy tumour cells. However, with complex biology in play, it is vital that meaningful data can be collected in vitro in order to better predict how effective a drug will be by the time it reaches human testing. The value of using complementary instruments together in order to achieve this data is highlighted on this poster. Here we describe a workflow combining live cell analysis and flow cytometry, enabling complementary parameters to be quantified. And to demonstrate the potential insight gained by combining these platforms, we have characterized two bispecific T-cell engager or BiTE antibodies. This combined workflow uses the Incucyte Live-Cell Analysis system and the iQue to both collect and quantify the data I'm going to talk through. The Incucyte Live-Cell Analysis system is a fully automated phase contrast and two-colour fluorescence system that resides within a standard cell incubator for optimal cell viability. It is designed to scan plates and flasks repeatedly over time. The iQue system is an advanced flow cytometry platform with a patented sampling method allowing for rapid sample acquisition to deliver fast actionable results. The ability to quickly analyse data is made possible with Foresight software, which provides analytics and results visualization tools that are not found in traditional flow-based software packages. Sartorius offers a suite of reagents and kits that can be used with these platforms for cell health and functional screening, and this workflow application also shows how some of these reagents can be used effectively across both platforms. This image gives an overview of the simple workflow which is used to connect the Incucyte and the iQue with a more detailed protocol being found on the Combined Workflow Applications webpage on the Sartorius website. On day 0, NUC-like green labelled target cells are seeded into a 96-well plate. The workflow has been designed so either adherent or non-adherent target cells can be utilised. Next, the activator of choice is added, and lastly, the label-free immune cells. Death dyes, such as a Nexin-5 red, can also be added here to monitor apoptosis. The immune cell killing reaction is then placed in the Incucyte, where changes in cell proliferation, death, and morphology can be monitored temporarily for up to 5 days. During this time period, daily cytokine samples can be taken from the plate, and this can be done aseptically in a cell culture hood by removing the plate between scans and sampling 10 microliters of supernatant for cytokine quantification on the iQue. Immune cell killing progress is monitored on the Incucyte system, and when an appropriate time point has been reached, for example when killing has plateaued, the cells are then prepared for analysis on the iQue system. This is achieved by a simple transfer step where the cells, still in co-culture, are incubated with AccuTase in order to lift them from the assay plate. They are then transferred into a V-bottom plate and labelled with antibodies for endpoint analysis on the iQue. All of the immunophenotyping carried out here use the Sartorius T-cell activation cell and cytokine profiling kit, or TCA kit. Using both instruments allows for the monitoring of dynamic changes in cell health, morphology and spatial metrics, as well as being able to choose an endpoint to then immunophenotype the cells and quantify cytokine release to provide a detailed account of the immune cell killing reaction. It is important to add that the multiple metrics outlined here can be achieved from just a single assay plate. In these validation experiments, the NUC-like green reagent mentioned in the previous image is compatible across the two platforms and so provides a useful tool to both validate the efficacy of the transfer between the instruments and also to cross-check results. In this first image here, we can see how the Incucyte software performs quantification of the green labelled target cells. The software applies a mask to the nuclei of the target cells, which is outlined here in pink. This produces a count of green fluorescent objects. The quantification on the iQue system is performed with Foresight, as shown in the dot plot here to the right. The NUC-like green signal is used to separate out the target cells and the unlabelled immune cells, so again, like the Incucyte analysis, a count can be performed. These next two graphs show the target cell numbers expressed as target cell count per image for the Incucyte and target cell count per SIP on the iQue, with SIP meaning the sample size taken from each well for analysis. The data has been taken from a single experiment where a nuclei count was performed on the last time point taken on the Incucyte before the same cells were then lifted and sampled through the iQue instrument. When these results are compared, we can see that the trends are comparable, where activated PBMCs resulted in a significant drop in target cell counts when compared to non-activated PBMCs or target cells grown in monoculture. So, although the two instruments use completely different methods of detection and quantification, this data shows that the two can confidently be used to study the same biology. A further example of how the two instruments can produce comparable data in terms of cell quantitation can be demonstrated here. A CD3 and CD3-CD19 BiTE antibody was used to activate immune cells in a target and PBMC co-culture. The first image on the left shows how a simple live dead cell dye was used to count live targets on the iQue. This information has then been displayed as a heat map using foresight to show a concentration-dependent relationship between the increasing BiTE concentration and decreasing target cell numbers. This iQue data was then used to construct a CRC to describe inhibition of proliferation. When this curve is compared to the Incucyte analysis of the same experiment, again looking at target cell numbers, the curves overlay and give highly comparable PIC50 values. This again shows the two instruments are sensitive to the same types of biological changes, which is important when using them to provide complementary data. This next slide shows how this concept can then be taken further to generate multiple metrics in order to better explain the complex biology that is in play during the immune cell killing process. These images taken from the Incucyte software show a green labelled SCOV adherent target cell line in co-culture with label free PBMCs. The PBMCs have been activated using increasing concentrations of CD3-CD28 dynabeads. As the time course progresses, at 36 hours the PBMCs have become enlarged, and by 72 hours target cell death is apparent, as described by the annexin-5 red signal seen in the image. And when you compare this to the 72-hour time point to non-activated PBMCs at the same time point, it is clear that in the non-activated wells there are a greater number of target cells present, smaller PBMCs, and little evidence of apoptosis. This analysis can then be further added to using the extra data collected using the combined workflow, which shows a detailed timeline of events for activation of immune cells and subsequent killing of target cells. Whilst this assay was running in the Incucyte, 10 microliter cytokine samples were taken from the supernatant every 24 hours and analysed using the iQue. Because the sample volume needed is so low, this step can be repeated many times with no effect to the ongoing experiment. Analysis shows that both interferon gamma and TNF, which are classical markers of T-cell activation, both increase in a concentration-dependent manner in response to treatment with the dynabeads. We see a significant increase occurs early in the process of activation at 48 hours, and literature discusses that the release of these cytokines by T cells is one of the early hallmarks of their activation. Effector cell proliferation was also quantified, again using the masking capabilities of the Incucyte. And at 72 hours post-stimulation, we start to see a concentration-dependent increase in effector cell growth. Only once immune cell numbers have significantly increased do we finally see the target cell numbers start to decrease again in a concentration-dependent manner. So if we view the data from the two instruments together, we can start to assemble an order of events which culminate in target cell death. This then gives a more informed picture of how activation and immune cell-mediated killing come about, and shows that this is not an instant process and one that occurs over time with multiple signals converging, all of which need to be taken into account. We then went on to characterize a CD3-CD19 BiTE antibody, which is a therapeutically relevant class of antibodies. BiTE antibodies have been engineered to contain two separate receptor binding domains, allowing it to bind to the CD3 receptor found on T cells, as well as a tumor-associated antigen such as CD19. When the BiTE binds to the T cell, it is able to initiate activation of the T cell, as well as binding the target cell expressing the specified antigen. This interaction brings both target and effector cell into close enough proximity so that the disease cell can be effectively killed. The CD19 antigen is overexpressed in a number of blood cancers, and here we used the non-adherent Ramos cancer cell line in co-culture with PBMCs. The Ramos cell line was again labelled with the nut green reagent to allow for identification by the Incucyte and iQue software. As expected, increasing levels of BiTE antibody induced a concentration-dependent decrease in target cell count. This effect was not seen with the control antibody. Changes in the BiTE-activated PBMCs were also interrogated using the Incucyte. Here we can see the difference in effector cell size between wells containing the BiTE and those containing control antibody. The presence of BiTE has caused the enlargement of the immune cells as well as their clustering. T-cell clustering is another hallmark of their activation, where they seek to round up the target cells before killing them. The Incucyte was used to firstly identify these clusters and then quantify them, and we can see that the average cluster area increased over time in a concentration-dependent manner, indicating that this morphological change was in response to BiTE stimulation. Again, from the same assay plate, cytokine samples were taken every 24 hours during the 3-day assay and analysed using the iQue. A concentration-dependent increase in the release of interferon gamma in response to BiTE stimulation was observed, as well as a concentration-dependent decrease in target cell numbers. We also compared BiTE-mediated killing to Dynabead-mediated killing. Interestingly, although stimulation with Dynabeads induced a much higher release of interferon gamma, this did not translate into lower target cell numbers. In fact, we see that a bead concentration of 75,000 produces nearly tenfold more interferon gamma when compared to the top concentration of BiTE-antibody. However, the BiTE induced a 93% reduction in target cells, compared to only 72% with Dynabeads, which was seen consistently across multiple assays. The cells were then lifted and labelled using the Sartorius TCA kit, where activation states from the T cells were compared between the BiTE and the Dynabeads. Both antibodies induce concentration-dependent increases in early, mid, and late-stage T-cell activation markers. Interestingly, we see that BiTE activation induces a clear left shift in relation to CD8, CD69 positive cells when compared to CD25 and HLA. Using the workflow to evaluate the BiTE against a broader CD3 activation has allowed us to tease out differences in the data, such as cytokine release and target cell death, whereby the specificity of the BiTE antibody allowed for more effective killing without a large release of interferon gamma, something which is important for developing immunotherapies so as to avoid cytokine release syndrome in patients. We have also used the combined workflow to characterize a CD3 HER2 BiTE antibody. HER2 is a solid tumour antigen which is overexpressed on certain breast cancers. This experiment has been carried out using an advanced cell model where the breast cancer cell line AU565 was grown as spheroids in order to stimulate a 3D tumour microenvironment. Here we show that the BiTE produces a comparable killing compared to the Dynabead positive control. However, when the T cells were interrogated with the TCA kit, no activation was observed. Dynabead activation resulted in the classical early, mid, and late T-cell activation markers. However, this could not be replicated with the HER2 BiTE over multiple experiments. This suggests a potentially different pathway or even different subpopulation of immune cells being involved in the target cell killing. This example shows the importance of being able to use the data from both instruments in order to be able to characterize drugs and predict how effective they will be. So by using the two instruments together, changes in cell function and morphology can then be related to T-cell phenotype and cytokine release. This approach has the potential to facilitate a greater understanding of the complicated biology involved in immune cell activation and killing of tumour cells. And the workflow described here allows the user to better incorporate multiple assays using a simple setup where all of the relevant data can be achieved from a single assay plate, saving precious sample and time whilst increasing productivity.